Today's lecture is about practical lecture is about soil sampling and preparation. While much attention is given to the laboratory procedure, the process of obtaining soil for analysis soil sampling is often ignored or poorly considered. Soil sampling actually uh, should provide a measure of average soil fertility level of the field and measure of how variable it is. We must be very careful while taking the soil sampling. Obtaining a representative soil sample from a field is a most important step for making a meaningful soil analysis. For soil sample collection, a sample should be composed of several subsamples representing seemingly uniform area or the field with similar cropping and the management history. Although there is no universally accepted numbers of subsamples for different field situations, normally 8 subsamples are taken per hectare in a diagonal pattern or randomly from a particular field, and then combining or mixing those samples into a composite sample. So composite sample should represent the whole field area. This is very important. So in this slide, if we look at this slide, uh, there is clearly mentioned here there are different types of the soil sample collection procedures. The most ideal one is considered the random method. If you look at there are the several samples can be collected randomly from the field, and these random sampling method is substantially saving in cost and the labor, and there is no universally accepted number of the subsample for different field conditions. As I already mentioned, we need to take 8 subsamples per hectare normally. And the time of the sample, we can take the sample. The soil samples can be taken anytime that soil conditions permit, but sampling directly after fertilization or the amendment should be avoided. That we must be very careful while taking the sample. The normal depth mostly 0 to 15 centimeter and 15 to 30 centimeter depth the samples are usually taken from, and 0 to 20 centimeter is a flow layer depth, and for normally the for the crop land area or field, if you want to take samples, then samples can be taken from 0 to 15 centimeter depth. For tree plantation or orchard, the depth can vary from maybe it can vary is go up to 2 meter. For if you want to study the nitrate leaching or any other leaching studies, then soil sampling depth may increase from 60 to 100 centimeter. The sampling tools normally uh, there are different tools available, and with different purpose. The samples can be collected through auger. If you want to take samples 0 to 15 centimeter or 30 centimeter depth, uh, then augers can be used. Soil sampling tubes are used if you want to take sample from deeper depth or up to 60 to 100 centimeter. Coarse samplers for if you want to sample from the soil surface, then up to 5 centimeter, then we can use core sampler. Farmer normally use shovels, spades. If the tools are not available, then the shovel and spade can also be used. And sampling process is very simple. Put after after taking the sample, you can put it in the plastic bag, and each samples should be properly labeled. When it's very important, if we don't label properly, then the all the efforts we do for the soil sampling go waste. And so it's very important to do proper labeling. For microbial studies, samples should be put in the freezers. Uh, After collecting the soil samples, we can take those samples back into the labs, and then put those samples in, spread those samples in the tray for air drying. And after air drying, we can grind the sample with pestle and mortar, or the automatic soil machine diagglomerators after removing the stones. That samples can be passed through, need to pass through with 2 mm sieve. 2 mm sieve is very important. And after sieving the soil, uh, we can store the soil in the plastic bottles or in the plastic bag for further soil analysis. Is a video demonstration about the tools we have in our lab. There are different tools which can be used, which I already mentioned. These kits are available, and different sampling tools. This one is the auger, uh, soil auger. Uh, as I already mentioned, is a very important tool for soil sample collection. There are different handles can be attached with the augers, and to reduce or increase its height. And there are different other tools available in this kit. uh, this kit is very important for soil sample collection. So uh, the augers, there are different types of the augers are available. If this auger uh, can be used for the, if the soils are a bit hard, then this can be inserted into the soil deep. And if you look at there the procedure, how we can take the soil samples from a different field. So after taking the samples, this, if the soil is wet, this type of the augers can be used. uh, these are bit uh, easy or feasible to use such type of augers in under different field conditions. So there are different types of the augers depending upon from which area you want to collect the samples. This video demonstration is about how we can collect the soil sample from a field. If we look at this video, we can easily uh, witness that if we don't have a auger, we can use a spade for the soil sample by digging a V-shaped hole. And if you look at this, uh, a V-shaped hole has been dug, and up to 15 to 20 centimeter deep. Then we can take a fine thick slice from smooth side, trim the slide, leaving the fine strip, then dump this strip into a clean bucket. We must have a bucket. We need to put the several random sample collected randomly from a particular field can be put into the bucket and then mixed all together. After mixing or making a composite sample, those can be put into a plastic bag, as we can see in this video. These plastic bags, after putting uh, those samples or the clots uh, into this bag, and after that labeling it with particular information uh, on this bag. This is very important. If we must need to label the sampling bag properly, we need to mention the name of that field, from which field you collected the soil sample, and also the depth is very important, from which depth you collected the sample. This must be properly labeled. And after uh, labeling and these bags can be then you need to transfer it back into the lab, where those samples will be further processed for further analysis. That's end for today's lecture. Thank you very much.